Hello, everyone. We are once again waiting for all of our attendees to join. Welcome, welcome. All right. Well, then let me start with the intro. So, live from Zurich and from Tokyo, in Japan. Welcome to our third webinar of 2025. And we are very excited for this webinar today. We just finished our first session earlier, and now we are in our second session. So we would like to thank very much our two guest speakers from Japan, Professor Yoshiho Ikeuchi and Dr. Tomoya Duenki for joining us. Hello, hi. Please say hi to our audience.
Hello. Hello.
Okay, so for today, we have a full program, and we will start with a short presentation from Muscle Biosystems by myself and by Sylvia. And we will have our main event from our two speakers from Japan who will talk to us about the construction and evaluation of neural organoid networks and circuits on high-density MEAs. So, all right, so then let me start with our introduction from Muscle Biosystems. So, our main topic for today from us is how to have experimental success in cellular electrophysiology. And just a hint, we're having something new to present to you for today. Discover more with the newest electrode technology. I'm not spoiling it, so we will talk about it later. But first, about muscle biosystems. If this is the first time that you're joining us, we are Muscle Biosystems. Thank you so much for joining our webinar. So we are a global technology leader in high-content electrophysiology, and we cater to hundreds of laboratories around the world, in 23 countries and counting. So we are based here in Zurich, Switzerland, and we have a dynamic team of around 60 people, 50% women from over 20 nationalities. And our devices have already been used by many users in over 200 publications, many of those in top journals such as Nature, Science, and so on. Also, our speakers of today have published quite high impact papers as well. And what our current focus is at Muscle Biosystems, oops, sorry for that, are the following. So we would want to actually provide you with the tools to be able to precisely analyze or do high content electrophysiology on the following biological samples. For example, 2D cultures such as iPSC-derived neurons and primary neurons, 3D samples such as organoids, spheroids, brain slices, and retina. So for the first part, Sylvia will be talking more about examples from 2D cultures. And from our speakers from Japan today, they will be talking more about organoids. So look forward to that. And the steps to ensure experimental success are as follows. So first, of course, you would want to fully functionally characterize your cells as much as possible. So they're very important. So you'd want to make the most out of your samples. Next, you would want to have reproducible results across many of your different experiments. Next, you would want to, of course, do these experiments with simple and intuitive assays. And, to go beyond, you would want to advance and access more from your samples by, for example, stimulation and other aspects in your experiments. So all of these are quite important, and this is what we're tackling today. So now I'd like to transfer the floor to Sylvia for more information about that.
Thank you very much, Mary. So I'm going to share my screen. All right. So I'm going to start this presentation discussing with you how to functionally characterize your cells. There are many techniques and technologies that you can use. For example, imaging, which gives you great access to the cell morphology, or patch clamp, which is the gold standard of electrophysiology and gives you insight on the cellular signals. And now imagine a technology that actually can combine these two. At Maxo Biosystem, we offer high-density microelectrode arrays. This is the next generation electrophysiology platform. You can culture hundreds of neurons and track them week after week, up to months. And not only you can have access to the whole neuronal network in parallel, but also have subcellular resolution access. For example, in this video here in the center, you see an action potential propagating from the axonal region, so from the somatic region, sorry, down to the axon and axonal branches. And in this way, you can get also information about the morphology of your cells. The field of extracellular electrophysiology also comes with some challenges. For example, you want to record from every cell, and this is very important for your research. However, traditional MEAs might have few electrodes and very spaced from each other. With our high-density microelectrode arrays, you have instead tens of electrodes that are dedicated to a single neuron, so you will not miss any part of your sample. Additionally, if you are, for example, in the field of iPSC-derived neurons or organoids, you may already know that it's difficult, specifically at the beginning of the experiments, to detect small action potentials. With our technology, instead, you have such a low electrode noise that it's possible to record even the tiniest action potential from your sample, and very early on. So the technology looks like this. You have in light blue these squares that are the electrodes, and you can appreciate how many tens of electrodes you have dedicated to a single neuron, here colored in orange. Going a bit more into details, at Maxwell Biosystems we have a one well or single well technology, but also multi-well technologies. The central part as the recording area is 2 × 4 millimeters square, and it has 26,400 electrodes at very high density. The size of an electrode is about 9 × 12 micrometers square, which is also more or less the size of a soma. Then the center-to-center electrode distance, also called pitch, is only 17.5 micrometers, and you have electrodes with very low noise. Additionally, each single one of these 26,400 electrodes is not only used for recordings, but also can be used for electrical stimulation. With our technology, as Marie was mentioning before, it's possible also to advance beyond the state of the art. Like, for example, was done in this paper published in Nature by the group of Magdalene Polimenido and University of Zurich, where they developed an iPSC-derived neuron cell lines to characterize neurodegenerative diseases, and to characterize this cell line, they used the Maxwell system. Or like the group of Feng Wu, where in this publication in Nature Electronics, they used brain organoids together with an AI tool and Max1 to demonstrate capabilities for speech recognition. And all of this was possible in this publication, and is possible with the Max1 and Max2 systems that we have. So the Max1 system is the one well dedicated system. It's versatile, flexible. It has the size more or less of a smartphone. It can go inside the incubator, and you can use it with our Max1 chips. Instead, the Max2 system is dedicated to higher throughput. You can put on it either 6 or 24 well plates. It has a built-in incubator that can control temperature and also CO2. With our technology, you can also obtain reproducible results. We have concrete solutions for consistent and long-term recordings. 100% of the products leaving our company to you are tested. We have more than 150 criteria that need to be fulfilled before the products reach you. We also have tons of protocols and video instructions developed with our partners and with our users. Here, I would also like to show you the reproducibility that you can obtain, for example, using a 24 well plate. Here you can find human iPSC-derived motor neurons, control line and 2-line modeling TDP43 mutations, and you have 2 conditions per cell line, and you can appreciate how, despite the different underlying activity in these samples, there is a high reproducibility in each column. Now you have a very powerful device. What you can also add to this is an all-in-one solution that is easy to use, that will allow you to record your sample and have insightful reports. This solution is called MaxLab Live, and it's our software. Will allow you to visualize your sample, record it, and then analyze your data, and also export it, the metrics, and also obtain reports. Just with a few clicks, you can localize your sample, check the baseline activity, and also study the connectivity and the network dynamics. You can go one step forward and achieve subcellular resolution, study the axonal function, morphology, and propagation of action potentials. And you can also stimulate your sample and characterize the cell excitability. Additionally to this, we also provide a simple and fast solution for high throughput experiments. We have an annotation tab that will allow you to easily organize your experiments. we just also recently released the batch analysis module to analyze all of your data simultaneously, and a report generation module to export your results ready to be shown. The results can be exported in PDFs or also PowerPoint slides ready to be presented, and also high-quality PNGs. Like, for example, in this application note that we just recently released in collaboration with Fujifilm, where human iPSC-derived neurons were used to model frontotemporal dementia, and in this case, a batch analysis module and record generation module were used. Today, I showed you how, with our technology at Maxwell Biosystems, it's possible to functionally characterize your cells. And this, it's possible because of the ultimate signal quality that we provide. We have the best-in-class high-density MEA with a very small electrode-to-electrode distance, low noise, and highly flexible readouts. You can also advance beyond the state-of-the-art. An example of this is electrical stimulation. We have single-cell stimulation that is enabled because of the small electrons and also low impedance. You can obtain reproducible results because of the optimized surface for culturing cells. You have a large area with more than 26,000 electrodes and many established protocols. And last, you can also run simple and intuitive assays that will bring you to an elevated user experience. We have an easy-to-use software, automatic data analysis, and export in batches. But we don't want to stop here. We want to go a step forward for you, and we want to constantly evolve. For this reason, we have a new chip dedicated to an improved user experience. Looking at the qualities of this chip, we have already the best-in-class signal quality with our technology. Now we go a step forward and we have a 50% increase in signal quality, meaning that you can detect even tinier action potentials. Concerning electrical stimulation, you could already achieve single-cell stimulation. Now you have a more effective electrical stimulation. With the same voltage that you were using before, you can excite more neurons, and the same neurons can now be excited with much lower voltages. Regarding the surface optimization, we have established protocol, scientific publication, but now we have also a new material on 43% of the surface that will enhance your cell culture viability. And last, regarding the user experience, to add on our easy-to-use software, we also have sterile chip and automatic chip ID readout, meaning that you plug in your chip, it's automatically detected, and all the relevant information regarding the chips are in software for you. And that's where we want to position ourselves. So for this reason, it's with great pleasure that I present you today the Maxon Plus. It's a newest excellence in electrotechnology. It's available to order now, and it features a new material for an enhanced performance. This material is called PDOT, and it has high electrical conductivity, it's biocompatible, it's mechanically soft, and also it allows you to have long-term stability. With the same electrode number and density, you have maximized performance and also an improved user experience. And with this, I would like to thank you very much for your attention, and I will hand over again to Marie for the rest of the talk.
Thank you so much, Sylvia. And yes, everyone, finally, we have launched our new latest chip called Max One Plus. So please try it. You can contact us directly if you want to see how it works in your lab. So yes, very excited about that. And now let's proceed to our main presentation for today, which is by our invited speakers from the University of Tokyo in Japan. Let me first introduce them before I have them here for the presentation. So first is Professor Yoshiho Ikeuchi. So after finishing his PhD studies on RNA modifications at the University of Tokyo, Professor Ikeuchi conducted postdoc research in molecular and cellular neuroscience at the Harvard Medical School and Washington University, St. Louis until 2014. After that, he returned to Japan, and he has been conducting research on functional integration and engineering of neural organoids at the Institute of Industrial Science at the University of Tokyo. So please welcome Professor Ikeuchi. Hello. And our second speaker for today is Dr. Tomoya Duenki. He just graduated actually in his PhD, so congratulations, Tomoya. So Tomoya is a postdoc researcher now at the Ikeuchi Lab, Institute of Industrial Science in the University of Tokyo. So he conducted there his PhD studies in chemistry and biotechnology. So he also holds a master's degree in biomedical engineering from ETH Zurich and focuses on engineering techniques to improve studies using neural organoids. So very much looking forward to your talk today, and the floor is yours.
Okay, thank you very much for having us today, and thank you very much all for joining. So we will talk about our attempts to construct the networks of organoid, because I think it's one of the biggest dreams for the neuroscientists to construct functional neural network outside a body. So for that, towards that goal, it's really exciting. A lot of research is going on, and it's really making a lot of progress recently. Sorry, okay, here. Oh, here. Okay, so, and just by looking at the some of the research coming out using like Max-1 system, there are many like fantastic research making progress. And here I'm just showing two recent examples. One is from Yamamoto and Hirata group in Japan, another from Bosch group in ETH Switzerland. And it's really clear that by having a group of neurons to form networks and then make bigger network between them, that can create much more functional network and complex activity that are sometimes shared between different groups, and activity going through different group of neurons that travels through very complex kinetics. And it's really amazing to see those works to realize the construction of functional networks. So we want to take a little different approach by using neural organoids. Here in the audience, probably everybody knows the neural organoids. I don't really need to go into detail, but I see that this is a very amazing way to create more complex networks of neurons, because when the neural organoids are generated from pluripotent stem cells, they go through these waves of differentiation into different type of cells, neurons and other cells like astrocytes. And then that creates like a microstructure that mimics the developmental brain that intrinsically creates a complex network. And you can create different organoids that mimics different regions of the brains that already gives additional complexity, but also you could assemble them to make so-called assembloids to mimic interaction between different regions. So that's all good. And then here's the one of the example of the recordings of the neural organoids, like cerebral organoid in this case, that are just placed on the Max-1 chip. And as you can see, these red parts firing action potentials. And then you can see that many different regions fired at the different timing, and sometimes like this all fire together, showing the burst, synchronized burst activity that really shows the intrinsic complexity of it. And that's really amazing to us already, but I want to bring your attention to the structure of the human brain. So as you can see, there are many regions, like including the gray matter of the cerebral cortex or other nuclei of the brain, that have a lot of cells. And in between, there's a big white matter that houses the tract of axons that are dedicated to connect different regions of the brain. And we think that's the key structural element that is the basis of the human brain function. And we would like to mimic it by connecting organoids. So that's what we're going to talk about. That's I want Tomoya to talk about in the next step.
Yes, exactly. So how do we mimic these interregional connections, these interregional axonal projections that we have in our brain? So what we first do is we generate these organoids, as you can see in the schematic on the left top. In this case, it was done with cerebral organoids. So after differentiating these organoids for 4 weeks, we will put them into a microfluidic chip that looks something like this in this picture. So this microfluidic chip contains two chambers and round chambers that you can see here and that are interconnected through a channel. And if you add these organoids inside, they will attach to the bottom of the surface and start extending axons in all directions. And however, given the physical restriction of the chip, the axons can only elongate in the direction of the channel. And if you culture them for a week or two, you can find that the axons will start extending axons until they reach the other end of the channel and eventually start connecting with the other organoid that is in the other chamber. And in this way, you can make reciprocal connections between the two organoids that are placed inside this microfluidic chip. And here on the left side, you have an example, or we have an example of GFP expressing cerebral organoids that were placed in this channel. And the first picture is like a picture right after we have placed the organoids inside. And already 1 week later, you can see that there are many, many axons extending from the organoid that also occupy the area where the channel is. And eventually, after another week, you can see that the axons bundle together into a axon bundle that will gradually grow in thickness over time. And so we were able to in this way to structurally connect these organoids, and we wanted to further confirm whether they are also functionally connected. And for this reason, we have placed these organoids onto electrode arrays. And here on the top left, in this image here, you can see the correlation of different frequency bands plotted against the time, where we compare signal of recorded from the left organoid compared to the signal from the right organoid. So we compare the signals recorded in these organoids, and what you can see is that initially you may not have too much high correlation in all kinds of frequencies. However, if you keep culturing them and keep them for another week, and when you start seeing that they are structurally connected, you can find that you will have a lot of overlapping frequencies and a lot of correlation, especially in the low frequency bands, when you compare the signal in the left and the right organoid. So in this way, we could confirm that they're also functionally connected. And what was even more interesting is that when you compare these connected organoids with single or with organoids that you fuse together, when you take a look at the activity pattern, you may find that the connected organoids display much higher burst frequency. They have more intense activity, as well as when you take a look at the burst interval coefficient of variation, you can find that this coefficient of variation gradually increases with time, indicating that you will have much more complex activity patterns or burst pattern occurrence with time compared to single and fused organoids, where you have a rather periodic burst occurrence. And so this was now done with was done previously with commercially available low-density array chips, but we have started to use more of the Maxwell Biosystem device and the system in recent years. And also with the HD-MEA, you can generate these connected organoids and can record from the two organoids that we connected with much higher resolution. And in this way, we can also capture a lot of the intrinsic complex activity that occur within the organoid, and also compare it with the activity and the synchronization between the two organoids. And with this, I would just like to briefly summarize some of these benefits when we connect organoids. So we can connect organoids while still keeping the complexity or the intrinsic complexity that is already within the organoid, but use this as building blocks and create even bigger, more complex structures. And by this, you will also integrate or introduce a new structural component, the axonal bundle, that will mimic also the structures that we have in our brain. And given that you have an axon bundle, you can also study the axon bundle itself. In fact, we have also developed other techniques to study and isolate the axon bundle in order to do downstream biochemical assays of it. And by connecting them in this way, you also have the organoids more compartmentalized. So it makes it easier to do individual manipulation of these organoids and have this modular network construction. And combining this with the HD-MEA chip, you can simultaneously get the information about the activity, which makes it a great tool for progressing the studies of organoid and neural circuits in our brain.
Mm-hmm. Yeah, so that's our so that's what the one of the examples that we've been using this MAX1 for these studies. But then we want to take it a little bit farther, I guess, like a little bit more. So one of the things that we wanted to ask ourselves was, how can we alter any functionality, if there is, of the neural network tissues that are made with the neural organoids? So that's what we wanted to do. But then we thought of, sorry, this one. we thought of many things, but then one of the things that caught our attention was this our development of the visual system. So the bottom timeline shows the how our visual system develops. And apparently humans open our eyes quite early on. As a human, like a baby, are born, they open eyes, try to see outside. But it's been said that it is very blurry at the beginning, but gradually it gains more precise sharpness. And as you can see on the right image, next example, the things getting more clearer and clearer as they every day see things that are existing outside. That really shows how those external stimuli go through the circuit, and that shapes the neural circuitry within. So that really is an inspiring phenomenon that our brain goes through. So we thought maybe organoids have to go through this also to gain any of the functionality. So that's our hypothesis. And also we thought probably to gain good functionality, the structure of the network is very important, and perhaps the functionality or functional assignment has to be done in the network. So we created some of the cerebral organoids that got cultured about 120 days so that it contains a good, relatively mature neurons and diverse neurons. And then we created circuit of one organoid, or two organoids that are connected, or three organoids that are connected. To do so, yeah, we are inspired by this research from Brain Engineer Group in UCSC, and where they place two organoids on MAX1 chip, and by that they allow axons to grow mutually, and that eventually forms very nice sparse connection, synaptic connection in between that shows the weak association or synchronization between them. So we thought that would be the ideal situation for this R study as well. So yes, so we placed them so we could see the activity. And I'm gonna skip that a little bit today, but we could actually see the axons grow towards each other, and then the activity synchronize weakly between them if we place 2 or 3 organoids. And then what we wanted to do was to set up a very, very basic functional task. So here we wanted to give 2 different stimulations and then ask if we can distinguish by looking at the response of the organoid without knowing the stimulation. We wanted to just simply use the machine learning algorithm as a classifier to evaluate the distinguishability of the 2 stimulations. So for example, what we did was we give 2 different stimulation at 2 different locations. Well, actually, the stimulation itself is the same, just the location of the organoid that received the stimulation is different, and we call it stim 1 and 2. If it's the case for 1 organoid, we gave 2 stimulation in 1 organoid and then recorded the output signals from the rest of the organoid. And then if it's 2 organoids that are connected, we gave 2 stimulation, so 1 and 2 stimulation 1 and 2 on the 1 organoid and recorded from another one. And then if it's the 3 organoid, we gave 1 stimulation on 1 organoid, another stimulation on another organoid, and then 3rd organoid is dedicated to the recording or the output. And then any output is fed into this simple machine learning classifier, SVM or CNN, and let it ask, let it check, let it guess which stimulation it was. And then we wanted to see, by giving repeated stimulation, if we wanted to see if that discrimination score goes up or not. And then to achieve it, we intentionally excluded the trials that give very high score from the beginning. And we intentionally chose a pair of stimulations that give very low score of discrimination, and then checked how it goes. So then, as you can see here on the left, the solo means 1 organoid, duo is 2 organoid, trio is 3 organoid. And as you can see, the beginning at day 1, the discrimination score is about 0.5, meaning that it is not discriminating. But then, as you can see, if we while we repeat the stimulation every day, and only the trio organoid gave higher score around day 12 and 13, 14, around that. And it only happened when we gave stimulation, but not when we didn't add stimulation. And that was the case for another algorithm like CNN. And as you can see on the left part, this score went up as we stimulated over and over again. That really means that the giving stimulation by itself could change the structure of the network tissue, and that really came out as a, in this case, distinguisher score as a readout of functionality. But the question is, how do organoids change the response to the stimulation? I would like Tomoya to explain a little more about it.
Yes, so after having seen that the organoids or the output organoids can start discriminating these 2 different kind of input patterns, we wanted to take a look at the underlying mechanisms or the actual response, how the response changed. And what you can see here on the top row, in the top row you can see the evoked activity in the electrodes before we did the actual stimulation experiments. And on the bottom, you can see how it changed after the time course of 2 weeks, how the evoked activity changed after 2 weeks. So what you see here is that the 11 × 11 electrode grid from where we recorded from the organoid, the output organoid of the trio. This is an example of the trio configuration. You may see that the circle size indicates the firing rate, the evoked firing rate, and you can see the same thing as a heat map on the left side. And you can see if you compare the stimulation 1 here compared to the stimulation 2 in the initial phase of stimulation experiment, you can find that they similarly evoke response in the left side of the organoid, as you can see here and here. However, if you stimulate these different kind of inputs over 2 weeks and take a look at the activity, the evoked activity after these 2 weeks, you can find that that spatially you can see in the map that the activity for stimulation 1 stays on the left side. However, in the stimulation 2, you may find that the activity starts to respond stronger in the right side of the organoid, and in this way we can, or maybe the algorithm can also decode better the 2 stimulated patterns. So this is how it looks on the map, on the spatial perspective, but we also did take a look at the temporal dynamics of the 2 stimulations before and after. On the top row, you can see the simulation response of before. Here on the left side, the network response, the network spike response over 700-800 milliseconds right after the stimulation. And you can see that initially they show more or less similar network response dynamics. However, after 2 weeks of stimulation, in the bottom row, you can see that you get a little bit more distinguishable response, in the sense that, for instance, with stimulation 2, you get a much sharper and shorter response, and also a stronger response compared to the stimulation 1, where it is a little bit longer, slower, more transient. So in this way, we could also locate from where this discriminality or this decoding ability comes. We could find that the actual activity also shows different patterns when we stimulate in these different organoids. We further did take a look at the alterations of the connectivity and the burst propagation between the organoids, or more specifically, the propagation of activity between input and output electrodes. So on the left side, you can see the occurrence of or the directionality of the activity that are propagated. You can see that in the trio configuration, you can find that the ratio of input to output activity is much higher compared to the output to input activity. And oh, I'm sorry, I forgot to mention, this is data after the stimulation. So we could find that after stimulating them repeatedly over the time course of 2 weeks, you can also introduce some kind of directionality into the circuit, indicating that there are some changes happening through the stimulation. And on the right side, you can see, this is again data of trio organoid only. You can find that we did quantify the time delay of the activity that was propagated. First of all, in B, you can see the time delay from input to output organoid, which seems to significantly decrease after we stimulated them for a long time. And if we quantified the propagation in the other way, so from the output to the input organoid, we could actually find that the activity propagation delay increased with time, indicating that the stimulation may have, through the stimulation, the network underwent some changes to have more efficient and faster signal transmission from input to output. So, and in this way, we could find that the trio configuration and the repeated stimulation that we can modulate the network, and that it is meaningful to connect multiple organoids. So what this raised in us our next question. So what would be the maximum number of organoids that we can fit on a Max1 chip if we wanted to go extreme and make large networks of organoids? And we took it a little bit as a challenge. So we tried our best to fit as many onto the Maxwell chip with the PDMS chip. And what we could manage so far is 18 organoids. So what you can see here, you have again the PDMS device that we attach to the Maxwell chip, and then 18 organoids in small chambers designed, and that are all interconnected through tiny small channels. And we also stained them and took images of them, and we could actually find that the organoids that we inserted into the device extended axons, and that they could form axon bundles between these organoids. It actually seems like the connectivity that we achieved looks much more complex than we expected. And we thankfully also conducted some recordings of these networks. And we could find that, again, here you can find different kind of activities within the organoids, but also from time to time you see these large bursts that recruit the whole network, leading into a big network burst, indicating that we can also achieve the functional connectivity between these small organoids that form these networks on the chip. So another reason why we actually wanted to push the boundaries and go extreme with many organoids is myself and our lab, in collaboration with Maxwell Biosystems, will actually have we will have a small exhibit at the Swiss Pavilion of the World Expo 2025, which is held in Japan, here in Osaka. Osaka in Japan. It will be for 2 months. The Swiss Pavilion has multiple rooms where they display different things, and one room showcases technological progress and Swiss technology. So we will have the opportunity there to showcase the Maxwell chip and our organoid networks that we plated on the Maxwell chip. And we will also have an event on August 4th. Maybe if you happen to be in Japan, please join. Yes.
I hope we convinced you the organoids, and especially when it's connected, it's a good it could be a good model for interregional brain network. And then because the network activity becomes different when it's connected, and functionality also can be unlocked by connecting and making some neural network. And I'd like to mention that the Tomoya did a lot of work, and along with Angela on the left, and then Foro, Soda, and Takuya also did a lot of work, and then other people from the lab also contributed a lot. Thank you very much for your attention as well. Thank you.
Thank you.
Thank you so much for the wonderful presentation. And I'm so happy we are quite synced today. So while us at Mass Biosystems, we are pushing the boundaries of MEAs with Max 1 Plus, you are also pushing the boundaries for the networked organoids with the 18 connected organoids. And this is really, really fantastic. Thank you so much. And now we are waiting for our questions here in the Q&A session. So first of all, we have Janusz Borosz. Hello, thank you for joining us this afternoon. Oh, you also raised hands, so yes. I think you have multiple questions. Would you like to ask them yourself? We can allow you to talk if possible. Would you like to do that?
Yes, can you hear me?
Yes, yes, all right.
Gladly asking. Wonderful talk, Professor Ikeuchi and Tomoya. It's really very nice.
Thank you. Thank you.
My first question is that when we do these micro tunnels on the chip, it usually gives us a factor 10 to 100 larger signals. So I was wondering why I never saw the communication between your organoids on the chip. What happens inside the tunnels? Why don't I see your axon bundles?
So there are actually two things. So first of all, thank you very much for your question. The example that I showed you was actually replated. So we created the connectoid in a different device and then added it on top of it. We actually can also record sometimes a little bit of activity in these organoids, but we also did recordings with the PDMS chip. And in this case, we can see in the edges, in the edges of the chip, we can find extremely amplified signals of the axons. However, I think in our case, the channel dimensions are a little bit larger. So I think the height of the channels are up to 100 to 200 micrometer, depending on the device, which is why I think we don't get as extreme amplification effects, especially in the middle of the channel.
And am I allowed to have another question as well?
Of course.
So this is then related to your stimulation protocol. You mentioned that you stimulated over many days, and then I was wondering how often and with what kind of stimulation amplitudes and protocols. So how was the stimulation that could achieve such a learning result?
Yes, thank you very much for the question. So we did daily stimulation for when we did the discrimination task. It was for like 100 seconds. We did 100 second stimulation 1. We let it rest for 100 seconds, and then we did 100 seconds of stimulation 2. And the stimulation itself was, if I'm correct, we did like 800 millivolts with a stimulation frequency of 100 hertz, 10 pulses. So we did 10 pulses, 100 hertz every second. So it would be 100 times. And
so I just wanted to add one thing. So we would like to do more if it's possible, but because we had to handle many samples and many stimulation had to be applied, so that was maximum we could do. So but if we can do more stimulation, perhaps it could maybe achieve faster score increase maybe, but this is what we have done so far. We haven't explored too much more on the more stimulation, which we would like to do as a next step.
Thank you very much for all the answers as well and for the wonderful talk. Thank you.
Thank you.
Thank you.
Thank you so much, Janusz, for the questions. Now we proceed with the other questions that we have. The next one is from Shinji Matsumoto. Thank you for the question. So he said, wow, 18 organoids. You could almost create 2 baseball teams with them. So anyway, will the 18 organoids start to synchronize their clocks, beats, and rhythms of spontaneous combustion of neurons over time? When they are super interconnected like this?
They do show network-wide synchronization over time. So you can find that they really burst in network, and it's like a wave that travels through the whole network. So I think yes, if that answers it.
But I guess also interesting question for us, because like we what's it called? We want to actually make sort of network that do not burst every we don't want all the neurons to fire at the same time. We want actually more like a gradual spread of the neuronal activity by creating a sort of bigger network, not bigger, what is it called? Like the like enough neurons to be in the organoids that create a complex network, and that are sparsely connected in between, that really creates the sort of big network size that allow action potential to travel through, action potential and synaptic transmission to travel through within the tissue, and that we hope actually to make them all a little less synchronized eventually, and that we believe that could be the foundation for the functional capacity for the network.
Thank you so much. So rather more complex activity and not just all synchronous activity.
Yeah.
Thank you. So next question from Wenji Ren. Thank you, Wenji. So can I ask, is the organoid cultured on the MEA chips directly, or you put the organoid on the MEA1 chip kind of at the time before you're recording?
Yes. So we do plate them. We have different kind of protocols. We for instance, with my 18 connected organoid, I plate them after 4 weeks of culture, and then they are chronically plated on it, and then I will record from week to week. Very similarly with the solo duo trio, we cultured the organoids free floating for 120 days and more. And afterwards we plated them onto the chip, and so it was not like immediate recording, but we kept them on the chip.
Thank you so much. All right, next question from I hope I pronounce it correctly, Woterreich or Wotrek. Thank you so much for the question. So he mentioned, thank you for your amazing work. My question, do you think connectoids will benefit more in understanding the disease mechanisms than assembloids? Ooh, good question.
That's a, I mean, question that I should ask, maybe. But well, so it depends. Of course, assembloids have been really pioneering the work of like interregional connections, and that's been really proven to model many diseases. And having axons is it's a very big difference between the system that we talked about and assembloids, which tend to be they're directly fused organoids. We think there, of course, direct and indirect benefits. Like for example, if you want to analyze axon formation, of course, we think it's better to have axons. For example, we have a collaboration with a group, a Novel Lab, and then so that they demonstrated the corpus callosum agenesis, like the hemisphere left and right cerebral cortex are lost in some patients, and that can be well modeled with the system because it has axons in between organoids, and that's really direct case. And we hope to explore more about other indirect benefits, especially by connecting different organoids and modeling a disease and analyzing signal propagation as well as plasticity. And yeah, we think we think this having different system like ours can benefit in many cases. So we would be very open to any discussion and collaboration opportunities. So yeah, please contact us also. Yes. Thank you.
Thank you so much for the answer. And yes, this is also a platform for you to start connecting with each other and building up collaborations. So we have another question from Andriy Domanskis. Thank you so much for the question. So why did you decide to switch to HD-MEAs from your initial experiments with low-density MEAs? What are the specific features of HD-MEAs that you need most in your studies?
That's a very good question. So, of course, the benefit of the HD-MEA is to have more electrodes. And for example, the solo duo trio discrimination study, we did the discriminator program, like algorithm, and that works well with the many electrodes that are placed under the organoids. But if it's just conventional system, we can only use like a few electrodes, and the algorithm cannot really run well, and simply because we cannot extract enough features that are within the response of the organoid. So that's the main reason that we feel that we strongly benefit from the HD-MEAs. That's what I think. Anything to add?
No, I think that hits very well the point that the more electrodes you have, the more information you have, and the more accurate information you also get. It can be also other simple or other things such as the propagation of activity within a small network, where you can get more information.
True. And then one thing to add is, like, I guess if it's max one chip or HDMI chip, you can place organize any place, and then you can record from there. Whereas the if it's conventional system, you need to place organoid on top of the electrodes, and then that defines the what's it called, structure, or what's it called, the architecture of the network that we create. So that's the main big difference. That really gives us freedom to design our experiment.
Thank you so much for the answer. May I follow up a bit? So was it also useful for you the stimulation part of the system?
Oh yeah, absolutely, right? We can choose any of the electrodes for stimulation, and especially we wanted to give two different stimulation, and that gives initially low score, and then we don't really know at the beginning, but like we were hoping that to increase, and then so like we have to choose, and we have to try and see where, first of all, where organoid response if we give stimulation. So we need to try and see where it's good, and then also about the score as well. So yeah, that was very nice that we could stimulate anywhere we want. So that's a really great benefit for sure.
Thank you, thank you. And now we have reached our last question. So for all of those who are still thinking of their questions, please feel free to contact us at info@mxwby.com just in case you don't have it now. But for our last one for today, this is from Scott. Thank you so much, Scott. Thanks for the talk. Since these are axons growing toward axons, I guess like this, do you think there could be communication between the axons? Have you confirmed that the axons reach the opposite organoid all the way, or is it just kind of, axon to axon in between? Quite curious question. Yeah.
Yeah. So then in development, there's like possibility that the axon-axonal gap junction form, but the most of the time we do see blockage of the activity propagation by blocking synaptic channels. So that probably means that the axon do reach the other organoid, and then it forms snaps with the dendrites of the receiving end of the organoid. And that happens both ways to create the functional connection between them. So we cannot really exclude the possibility that there are axonal connections. But for the most of the part, it is like regular synaptic connection between the axon and dendrite.
Thank you. Thank you so much. From Bram Service. I hope I pronounced it properly. So thank you so much for your question. How reproducible are the findings? Do you think it is related to the specific iPSC line or neuron type? So I think this is for you now, Ikeuchi Sensei and Tomoya.
Yeah, thank you very much for the question. We take the reproducibility seriously. So we have done the same work with two different cell lines. Not more, we would like to do more now, but we also have done this with the cerebral organoids. So that's a particular type. So what we have done, and we have done many times, and we of course take the statistical power, carefully. And for our trial, this is one of the things that we can say this is reproducible, because it's true that many things are variable, but we take good care of how we manage the data setup and how stimulation gives the initial value and those things. And with those careful consideration, it becomes more reproducible with the experience. Yeah.
Thank you so much for the answer. So I just wanted to mention again to our audience, yes, you can use the chat for just saying hello to us, but please add your questions to the Q&A session so that we can see them. Okay, so the next one is from Jessica. Hi, Jessica. She says, hi, thank you for the nice talk. Maybe I missed it, but I saw that you can register signal not only from the axons, but also from the whole organoids. I just wonder how big are your organoids and how old are they?
So the size wise, actually so as you know, the organoid can be very small to very big, and if it's big, we cannot fit many organoids on the chip. So we actually control the size of the organoid when we produce it, as well as the later on we can slice and make it smaller as well. So typically we for this one, solo duo trio, we have about 600 micrometer in diameter, and that's 5 to 600, I guess. And that's that we use. And then what was it? Sorry, the other question was
How old are they?
how old are they? We culture about 120 days, 100 to 120 days for this work because we want to have a good variety of neurons, including the upper layer neurons. So that's what we did for this one. Sorry, we didn't really include that information during the talk. Yes, thank you for asking.
Thank you so much. Maybe just to follow up. So after 100, 120 days, that's when you started to connect them. So they were not connecting beforehand. They were individually cultured.
Mhm.
And started
Yeah,
connecting.
yes.
Okay, thank you.
For the solo duo trio project, for instance.
Yes, and then for 18, I guess it's a little different story.
Yes. So the slides that I showed you at the end, for instance, since I wanted to add as many organoids as possible, they're actually much more smaller because each of these chambers are only like 400 or 500 micrometer in size. So they are much smaller, and I already introduced them up like for 4 weeks of culture.
Thank you for sharing, Tomoya. So we have more questions coming. So this one is from Pluni. Hello, Pluni. So how can the analysis of connected organoids be translated to the understanding of functioning organs eventually? What is still required for this translation?
Translation as in like
For
understanding.
understanding of connected organs. So how can you kind of use the connected organoids to understand functioning of connected organs eventually?
Yeah, understand the function of organs eventually. So I guess maybe this could include a lot of different things. Well, as in like, of course, our organoids by itself, but also like connecting them on chip outside of body is not brain. Like if you're not really making brain, so we need to be careful about it too. But at the same time, what we're trying to achieve here is to extract some essence of the structural element and correlated functionality or change of activity of functionality due to the network alterations. Like here, we're connecting the organoids. Like we see that as a modular network inside. We connect them and ask how that would change the overall activity. And that is for us also it's a little interesting task to do because it will not gonna make the whole brain. So we would like to understand part of the element or what is it called, like essence of how it could operate as a system from the bottom up approach. So that's what we're doing. But along the way, probably we need to do another translation, probably using like disease models and such to understand if it's reasonably recapitulates the disease deficits. And if that's the case, we could also say that the model is right in some way too. So that also we need to do more on it. So.
Thank you so much. Actually, my curiosity, not here in the questions. Do you have any insights why the connectoids are much more complex compared to just fusing the organoids together?
Yeah, that's a very good question. So we think the network, like structure, should be what's it called? Should have some sort of sparse nodes and connections to make the network like bigger. So I suppose that the organoid is good and complex, but it is in a way too well connected within. And there are too many synapses, and therefore, if you poke or stimulate any places, I guess it could still burst, fire synchronize everywhere, and which is not the case for the brain. I guess brain has a big network, and different parts can be active, and different timing, and to allow that, we need to create a bigger network size, and for that, I guess it's good to have a not fused, but purposefully separated but connected to create the connection of the organoid connector.
Thank you so much. Very interesting. So continuing with the questions from our audience, this one is from Mitsuri Ishikawa. Thank you for the question. What are the appropriate strategies for normalizing the degree of inter-organoid connectivity, particularly in terms of accounting for variability in organoid size, cell number, and projection density? I think this is a common question for all the organoids researchers out there.
Yeah, it's very difficult. I guess we really need to take good care of it. Like, what do you say, Tomoya?
In terms of, so I showed you, for instance, just before one plot that was like the so-called wavelet coherence, where you can correlate like the signal, the actual raw signal traces of two electrodes in a time frequency dimension. But there are also other ways, such as looking only at the spiking activity of the organoids, and take a look at how the spiking activity develops over time. You can do it with histograms, for instance, and then again correlate this histogram or the spiking activity of between two organoids. When it comes, however, to trying to normalizing the degree, I'm not sure, or I haven't really done or tried to do normalization. So far, I did a lot on absolute values. What you could do, for instance, is, I don't know, maybe you can normalize it with the correlation within the organoid. So you can make like intra-organoid synchronicity compared to the inter-organoid synchronicity. I haven't done it, but maybe it makes sense.
Yeah, it's true. So for at this moment, I guess we're trying not to normalize too much mathematically. We try to make the even tissue right now, but yeah, for the future, I guess we will need to vary, have more variable set up and see what how it changes and all those things. And of course, the normalization could be very, very important topic. So we would like to explore more for sure. Yes.
Thank you so much.
That's what we
Will
have.
be our last question for this session. If for those who would like to ask more questions, please feel free to ask us by email later on. But this last one is from Kagistia. Hello, Kagistia. Thank you for your question. She said, "Thank you very much for the excellent talk. I'm amazed by the recording of the 18 organoids. They are very beautiful." Yes, indeed. So she's wondering, what kind of stimulation did you do for the single, double, trio assay? Thank you so much.
Yeah, so other than like giving two different location, the stimulation itself was the same, but we gave 10 pulses of the same like square pulses of the same stimulation, like intensity stimulation to the organ, and we made sure that triggers response, and then use those. Yes.
All right, thank you so much. All right, well, this has been a really fantastic webinar. So thank you so much, everyone, for joining us. I just wanted to mention that our Max One Plus chip with neurons are actually also available as postcards. So if you get them, for example, from our exhibitions or from our booth, please feel free to share with us your pictures that you take with them around the world, and we would be happy to post them on social media and also in our presentations like today. And meet us also in person in our upcoming events. So we have ISSCR annual meeting in Hong Kong coming in June. We have the MEA meeting in July, and we would like to also mention that our Maxwell Summit is actually happening as a Maxwell Symposium together with the MEA meeting this year, and we already have our confirmed speakers right here listed. So looking forward to seeing you there. And if you want to know more information and to register, please see in our chat for more information. And yes, so this is going to be on July 8th, and registrations are open, and we also have travel grants, so please join. And finally, these are all the next events that we are joining in the next few months. So if you're in the area, we are happy to meet you. And once again, thank you so much for your kind words about this webinar. Thank you so much for joining us. And thank you so much to Professor Ikeuchi and to Tomoya for joining us all the way from Japan, now at 1 a.m. In the morning, and to Sylvia together with me today from Muscle Biosystems, and to all of our organizers of the webinar. Thank you so much. See you next time for our next webinar, and happy to see you today. Thank you.
Thank
and
you.
goodbye.
Bye-bye.
Thank you. Bye.
Bye.